So the next thing we want to figure out is how we can actually use that DNA to help direct our cellular activities. Just like the information stored in a textbook is functionally useless until it is read and put to work, the information encoded in DNA is worthless until the information is read and acted upon. Genetic information is put to use by the production of proteins. DNA is read and transcribed into RNA. The RNA then controls the primary structure of the protein. Does anybody remember what the primary structure of a protein is? It's going to be the sequence of amino acids. So the significance for DNA is that DNA codes for RNA and RNA codes for the sequence of amino acids that is ultimately responsible for the shape, structure, and function of the proteins. And proteins are everything. Proteins are the structural units of our cells. Proteins are involved in our enzyme activities. Proteins regulate metabolism. Proteins are so essential for everything in life. And we monitor life, or we regulate, the formation of those proteins through the information that is encoded in our DNA. So one thing that I'd like to kind of point out as we're talking about DNA and RNA is that when you are reading your textbook, you do not sit down and read the whole thing cover to cover and go about your day. No, you skip around and you read the sections that are relevant to the portions we're reviewing in class. Now, in this particular class, we are actually going to be reading the entire textbook, and we are going to be doing it in order. But I hope that you're not sitting down and trying to read the entire textbook cover to cover. Instead, what you should be doing is focusing on the sections that we are going to be covering in our class, focusing on the chapters one little bit at a time. DNA and protein synthesis kind of occur the same way. DNA synthesis, we sit down and we copy the entire textbook cover to cover because that's what we want to do. When we are making more DNA, we want an exact copy. We don't want to interpret that. We don't want to try and understand it. We just want a literal exact copy of that DNA for the processes of DNA synthesis. But in the case of RNA synthesis, in the case of protein expression, if what we want to do is make insulin, we're not going to sit down and read our entire human genome. We're not going to unwind and read all 23 chromosomes just so that way we can figure out how to make a little insulin protein. No, we're going to go to the portion of our DNA that encodes the information for how to make insulin. We're going to look at that tiny portion, copy down notes, and then use those notes to make proteins, to make that insulin. So when we are making a copy of DNA for mitosis, we're going to sit down, copy that strand of DNA from one end to the next. But when we synthesize proteins, we only want to read, copy, and synthesize the portion of DNA that codes for that particular protein. This is what's known as a gene. So the gene is going to be the segment of DNA with instructions for producing one particular protein or one polypeptide chain. When this protein is produced, we say that the gene has been expressed. So you're going to hear that term a lot, genetic expression, DNA expression. When we are expressing a gene, it simply means that we have turned that DNA into a protein. So we're going to do this through the aid of RNA. In order to really understand how this process works, we first need to take a look at what RNA is. RNA, also known as ribonucleic acid, is going to be made up of nucleotides containing the sugar ribose, and then one of four different nitrogenous bases. So just like with DNA, we have a phosphate sugar backbone. And we're going to have our nucleotide bases we're going to have our nitrogenous bases sticking off to the side. Now the four nucleotides that we have in RNA are going to be adenine, cytosine, guanine, and uracil. Uracil replaces thymine. So in the case of DNA, we have adenine, guanine, thymine, and cytosine. In the case of RNA, we have in RNA, we have adenine, cytosine, uracil, and guanine. Thymine has been replaced by uracil. Now, the complementary base pairing is still going to occur. When we are copying down our information from DNA into RNA, we're still going to follow follow our complementary base pairing rules. But in this case, although cytosine still pairs with guanine, adenine is going to bind with uracil. Now, RNA is going to be single-stranded. Unlike DNA, we don't have a double helix. we just have a single strand of DNA with our phosphate sugar backbone and our nucleotide bases sticking off to the side. Now, there are a lot of similarities between DNA and RNA. Both are nucleic acids, both are composed of nucleotides, both have that alternating sugar-phosphate backbone, and are made up of 4 different types of bases. However, there are a couple of key differences that are very important to our understanding of RNA and its function. DNA is really only ever found in the nucleus and in the mitochondria. Our DNA is our hard copy instructions for life. We do not want to mess that up. We don't want to risk anything happening to our hard copy. So the DNA is not going to leave the nucleus. We're going to keep it housed safe and sound inside of the nucleus, except for during mitosis. During mitosis, our nuclear membrane is going to dissolve and our DNA is going to be exposed. So before that happens, we wind our DNA up very tight and we condense it down into chromosomes to help protect our DNA during the process of mitosis. So DNA is found in the nucleus and there's a little bit found in the mitochondria. RNA can be found pretty much anywhere throughout the cell, especially anywhere where ribosomes exist. DNA is going to be responsible for storing genetic information. RNA assists in the processing of genetic information. RNA is going to be your notes. So your DNA, I like to think of the DNA as my hard copy textbook. My RNA is my notes, my notebook. I take notes, I scribble notes in my margins of my notebook. I rip out pages that I don't need. I take pages that I do need with me wherever I go. It's going to be much more fluid and it's just helpful in the process of studying and processing the information that I learned in class. Now, the main sugar of DNA is deoxyribose. That's where the D comes from in the DNA. The main sugar in RNA is going to be ribose. That's where the R comes from. The bases found in DNA are adenine, thymine, cytosine, and guanine. The bases found in RNA are adenine, uracil, cytosine, and guanine. DNA is double-stranded, where RNA is only single-stranded. DNA is going to be transcribed to produce RNA. RNA is going to be involved in gene regulation and is translated to produce proteins. Now, there are 3 major classes of RNA that are going to be involved in protein synthesis. The first type is messenger mRNA, or messenger RNA, which, like it says, it's going to be our messenger. It's going to take a message. It's going to copy down the genetic information from our DNA, take it out of the nucleus to the ribosomes for the synthesis of the proteins. Like I said, we want to keep our DNA, our hard copy, as pristine as possible. We want to avoid any sort of damage or mutations from occurring to our hard copy DNA. So our hard copy DNA sits inside the nucleus nice and safe. Now, unfortunately, we can't turn that into proteins. Do you remember who are considered our protein factories? Where's the site of protein synthesis in a cell? The ribosomes. The ribosomes are where DNA synthesis occurs. I'm sorry, not DNA synthesis. It's where protein synthesis occurs. So we have to somehow get the genetic information that is bound up inside the nucleus out into the ribosomes. We do this through messenger RNA. Messenger RNA is where we copy down, we go into the nucleus, we copy down the information we find inside the nucleus, we take it out of the nucleus, we take it to a ribosome, and then we synthesize that information into a protein. The process of copying down the message to send to the ribosomes is called transcription. So transcription is the process of transcribing DNA into RNA. Ribosomal RNA is the second type of RNA. This is going to complement with ribosomal proteins to help make up the ribosomes that are going to synthesize the proteins. So ribosomal RNA is simply part of the ribosome, and we'll take a look at that a little bit later. Transfer RNA is going to read the message from mRNA and assemble the correct protein sequence. This process is known as translation. And the reason that we call it translation is because we are translating a sequence of nucleic acids into a sequence of amino acids. So you can kind of think of it as like our translation from, here we go, here. We'll start off, the transcription of DNA to RNA, it's kind of like transcribing down a conversation. You write down a conversation, you write what you hear into words. Translation is turning those words from Spanish into English. We're translating from the language of nucleic acids into the language of amino acids. And in this way, we're going to synthesize our proteins. We're going to spend quite a bit of time on transcription and translation in our next section, so that should be sufficient for now, but I just wanted you guys to be familiar with the different types of RNA that are going to be involved in this process. All right, so you should be able to compare and contrast the structure and functions of DNA and RNA, and explain that term, semi-conservative replication of DNA. Why is DNA replication semi-conservative? Why do we use that term? You should also be familiar with the major steps in DNA replication and the enzymes that play a significant role in DNA replication. I want you to be familiar with the role of DNA helicase, DNA polymerase, and DNA ligase. So please review those, helicase, polymerase, and ligase. And then finally, you should be able to explain the roles and the significance of mRNA, tRNA, and rRNA in the cell. We are going to spend quite a bit of time dealing with these in our next section, so if you are not yet confident with that, that's fine. But by the end of this chapter, by the end of this chapter, you should be able to explain the role of mRNA, tRNA, and rRNA in a cell. All right, in our next section, we're going to talk about the process of gene expression. In other words, we're going to talk about transcription and translation.